Everyone. Today I'm going to be presenting my project, which is on the probabilistic description of patient anatomy for the predictions of treatment-induced toxicity. My name is Carl Grover, and the supervisor for this project will be Martin Ebbett. So to begin with, for treatment-induced toxicity, this is defined as the side effects which result from the irradiation of healthy tissue during radiotherapy. It is one of the limits for the effectiveness of radiotherapy, as it is inevitable that we will end up radiating healthy tissue during the process. For some statistics, about 20 to 25% of patients report adverse physical consequences of the treatment, which negatively impact their overall quality of life. For pelvic cancers in particular, gastrointestinal toxicities end up developing in about 10 to 20% of these patients post-treatment, and 20 to 40% of these cases end up reporting moderate to severe impact on the patient's overall quality of life. This ends up placing a health cost burden and an overall inconvenience on the patients post-treatment. So for the conventional approach to radiotherapy, it accounts for this by considering it as an optimization problem. So we want to maximize the overall dose that is delivered to the localized tumor volume and get good dose coverage without compromising the overall adjacent tissue, healthy tissue, and causing treatment side effects. This relies on the three-dimensional characterization of the patient anatomy using radiographic imaging or other imaging modalities in order to predict the dose that is delivered to different tissues for a given arrangement of radiation beams. This is creating what is known as the treatment plan. And for this, predictions of the treatment efficiency and the treatment-induced toxicity end up being used for the optimization of a treatment plan. In order to use this process, we end up producing a simple vector representation of the patient anatomy using delineated structures. So for the diagram on the right, we can see that there is a contour of the prostate, and so they have defined the overall structure of it by delineating the structure. And the same has been done with the rectum, which is the organ at risk in this case. So by doing this, it ends up discarding a large amount of the spatial information that is provided by patient imaging. And in doing this, it ends up limiting the treatment with a couple of drawbacks. So one of which is that in order to make the toxicity predictions using the delineated objects, they need to be made according to dose volume models, such as the dose volume histograms. And why that isn't exactly ideal is because they end up displaying spatiotemporal degeneracy, which means that multiple dose distributions are capable of giving the same DVH. So while one may have a very concentrated dose at one section, another may have a pretty even spread, but still give the same DVH in the end. And because of this, it doesn't really account for any of the localized differences in radiosensitivity. So it is pretty hard to be able to assess the overall, it's hard to assess the overall ability of the efficiency of the treatment by not considering the spatial information that's provided. As well as this, it also relies on a flawed assumption that the patient anatomy is capable of being precisely defined. In reality, it ends up being subject to a number of uncertainties. This can include the spatial uncertainty that exists in the imaging modality that's used, a temporal uncertainty in the fact that the image is being taken at a single point in time and may not necessarily reflect the patient's anatomy during actual treatment. And as well as this, we also have the inter-observer delineation uncertainty. So what is this? So this is the result of the fact that two different observers will end up producing two distinct definitions of the same anatomical structure. And because of this, we'll end up having two different treatment plans being produced. So for the diagram on the right, this is delineations of the gross tumor volume for primary esophageal carcinoma. And there was 5 different observers that ended up delineating this. And you can see from image B that there's quite a wide variety in the delineations that are produced. And this variability is one of the most significant sources of uncertainty in modern radiotherapy. Factors that can influence this include the experience and familiarity of the observer with the particular cancer they're working with, the formal education which they receive, particular guidelines and training that are in place at the clinic that they're working at, as well as the fact that they are two different individuals. They're going to have different biases on their defining of the anatomy. So a means that physicists have come across to be able to better incorporate the spatial information that's provided by the CTs is to consider spatial models for toxicity prediction. And here I've listed a couple of them. So here we have the dose surface mapping, there's dosiomics, deep learning methods, and the one which I want to focus on today is probabilistic treatment planning. So Shosharina in 2018 ended up offering a more flexible definition of the clinical target volume with the clinical target distribution. And what that was is that it ended up representing the clinical target volume as each voxel in the patient being assigned a probability that ended up containing tumorous cells. This, along with other studies in the area, ended up providing a lot of benefits in using this approach. These include the fact that there was an ease of testing alternative treatment plans without the need of completely recreating the CTV. As well as this, it was found to be able to be incorporated into the clinical treatment planning approach. So Shasharina in particular ended up finding that they could implement it very easily into the current approach compared to other spatial models which have a little bit of a harder time with being able to be implemented. And most importantly, it was able to incorporate uncertainties for the target volume delineation. So it provided a means to be able to more effectively account for the inter-observer delineation uncertainty by considering a probabilistic description. As of currently, however, the research that is currently on this is limited to the target volume only, and there's a lack of publications which talk about the probabilistic description of the organs at risk. With this in mind, this builds into what my project will be about. So the overall aim of the project will be to construct a voxel-based framework for the toxicity prediction that represents the patient anatomy as a three-dimensional probabilistic map. This will be able to account for multiple observer delineations to have the model be more robust to the inter-observer delineation uncertainty that's seen within the current framework. And in particular, when we refer to a probabilistic map, what this means is that each voxel will contain probabilities that it represents a particular structure. So we may have that, given the multiple observations that were created for the structure, there is a 90% chance that this voxel may be the rectum, and so on, and be able to represent the entire patient as a probability map. So before we continue, I want to take a moment to just address one of the methods that will be used, as well as the data sets we'll be using. So one of the methods that will be used throughout this project is atlas-based auto-segmentation with weighted voting. So this is a method that allows for you to automatically generate contours based on a reference atlas. This uses deformable image registration, and as we look on the diagram on the right, we have a reference image. This ends up being transformed and compared to the target image, calculating similarity metrics. And then using optimization, it alters the transformation and it goes through this entire cycle for the deformable image registration. Following from this, the transformation is taken and combined with the reference contour in order to for contour mapping. And for each of the reference contours that are created, they each become weighed based on the mutual information provided between the reference image and the target image. To weigh them, they're combined to create a final target contour. For this project in particular, we will be using instead of a reference contour, we will be having reference probability maps, and they will be combined to create a target probability map for the data set that we'll be using. So for this, there will be two different data sets. The first one will be a 5-patient cohort of prostate cancer patients. And in particular, each of these patients have had structures delineated by 10 different observers. So in total, we'll have 50 delineations to be able to work with. And for these, we'll generate probability maps, and these will comprise of the atlas to then generate probability maps for the second data set, which is the CHIP trial data set. So this stands for the conventional versus hyperfractionated high-dose intensity modulated radiotherapy for prostate cancer trial. It was conducted by the UK Institute of Cancer Research and contains 3,216 patients with full treatment information provided, including CT, the different toxicity calculations for genitourinary and gastrointestinal, quality of life data, and a lot of others that will be used later on the project to evaluate the overall performance of the framework. So for this, we'll be generating the probability maps for these patients using the atlas-based auto-segmentation, with our reference patients being the 5 from the earlier data set. The project can be broken up into 4 different parts. So the first part will be to develop a method for generating anatomical probabilistic maps, and these will be using the multiple observer delineations that we have for the first patient data set. To begin, we'll need to review the literature in order to establish a few requirements and precedences that will be required for creating these probabilistic maps. In particular, we can expect that the current publications that exist on probabilistic descriptions and applying that to radiotherapy will likely serve as a very good foundation, even though they only cover the CTV at the moment. Following this, we'll then describe a formalism for the anatomical probabilistic maps, and then be looking to create them for the first patient data set. Following this, we will be deriving probabilistic maps for the CHIP trial cohort, and this will be utilizing the atlas-based auto-segmentation. So for this, we'll first develop a pipeline for the propagation, and this will involve needing to first process the CHIP data. So this will include removing the treatment bed from the images, as well as ensuring that they have a consistent superior and inferior range on the scans, as well as the orientation that is all matched up. Then after that, we'll perform the atlas-based order segmentation with the weighted voting. And previous work has been done by Angel Kennedy on this, with a model being developed in our own study, and it is likely that she will be of sufficient assistance with being able to properly implement and get this working. Following this, we'll be taking a subset of the CHIP trial cohort to be used and taken forward with the project. This is mostly because of the computational cost, and it isn't ideal to really be taking forward the full 3,000 patients, so we're going to select a certain subset and take those through. And using those and the auto segmentation method, we'll be generating probabilistic maps for the CHIP trial subset that we create. The third process will be to develop formulations of the toxicity metrics that incorporate probabilistic maps. So for these, we'll be taking predefined toxicity models and figuring out means to translate them so that instead of using discrete descriptions of anatomy, they can use a probabilistic one. So again, we'll need to review the literature in order to assess a number of toxicity metrics and find if they would be suitable for having a probabilistic description applied to them. Some of the models that can be considered will be dose volume models such as DVH, normal tissue complication probability models, and as well as this, we can also consider spatial models such as the dose surface mapping and dosiomics, and probably be able to implement them into the framework that we are creating. So once we've selected a few models that we'll be using, we'll then need to derive methods for calculating toxicity responses by using the probability maps. So this will be building upon the previous calculations that are made for binary maps and transferring them over to consider a probabilistic representation of the anatomy. For the third objective, we will then be using these models that we create to calculate toxicity metrics for the CHIP trial cohort, and this will be using the probability maps that we end up generating from the previous part. Finally, in order to assess the overall performance of the framework, we'll be comparing the toxicity predictions that are calculated for the probabilistic representation with the discrete descriptions of patient anatomy provided from the original CHIP cohort. So for this, we'll be conducting a statistical analysis to compare, and this will be an in-depth analysis of our novel approach that we are considering with the conventional approach. And with this, we can expect to use a number of statistical methods, and one of which will probably be t-tests. And yep, so we'll use this to overall just assess the performance of our framework and see how it compares to the conventional approach. Some of the expected benefits that we can expect from this project is that we'll hopefully provide an improved framework to be able to improve upon treatment-induced toxicity predictions by considering the spatial information that the current framework currently lacks. In addition, one of the downsides of the current spatial models that we considered, like DVH and dosiomics, is that they cannot be readily implemented into the current planning paradigm that's used clinically. And by using this framework and the potential to incorporate these models into the calculations with probabilistic representations, it could provide potential for these models to be considered clinically. And as well as this, it'll be building upon the gaps in the literature that are currently exist regarding the probabilistic approaches to treatment planning, with most of it being focused on just the CTV. With this, we'll be able to extend it out to be considering all structures within the patient. And just to conclude, I've constructed a research plan that basically summarizes what we're doing, just in case it was a bit hard to follow. So we'll be taking the 5-patient data set and producing a probabilistic maps for these. Using these probabilistic maps and atlas-based order segmentation, we'll create probabilistic maps for the CHIP data set. From there, we have toxicity predictions models, and we'll be formulating means to be able to translate them to be calculated with a probabilistic map instead of discrete definitions of anatomy, and be applying these to the probabilistic maps created for CHIP to determine toxicity metrics using probabilistic anatomy. And then finally, comparing the discrete anatomy toxicity metrics and the probabilistic anatomy toxicity metrics to get an overall comparison of the performance and see how the framework performs. Thank you for your attention. Is there any questions?
Great. Thanks very much, Lauren. Well done. Nice presentation. Excellent. So any questions from Corey?
Yeah, I have a question.
Yep.
Andrew?
Could you go back to your summary slide, number 19?
Yep, not a problem.
Okay. So from the, I just want to make sure I get this right. From the atlas, how much data is in that from the CHIP probabilistic maps? Like after you apply your weightings and so on, how many patients is in the atlas? data set?
So in the Atlas dataset, that's consists of the 5 patients. So in total, there's going to be 5 patients within the Atlas. We're going to using the Atlas-based auto-segmentation, do deformable image registration to get the probabilistic maps for each of those 5 atlases, and using weighting, combine those to get the overall probabilistic maps for a CHIP patient. As for how many of the CHIP patients? Oh yeah.
So how many patients are in the CHIP trial data set?
So that was 3,000. That was about 3,000 patients, 3,216. Okay. Yeah.
Yeah, because I'm just wondering if you see like any problems coming up, or if you have to account for the fact that like if you're basing it on say 5 patients and 5 outlines or that kind of thing, or 10 outlines on 5 patients, there might not be as much like variation as in like a 3,216 data set. I can imagine there'd be maybe some different type of cases, and I'm wondering if you think that would affect the results whatsoever, or.
Yeah, well, it's definitely kind of hard to know.
Yeah.
Yeah.
Maybe if there's some way to see if some really different type of patient data sets in that chip trial set, if perhaps the models, or not the model, but this kind of system's performing worse on those, and if you remove those, if it would perform better overall. I'm not too sure, and I'm not sure if I understand the whole entire process exactly.
Yeah.
I have this really, really good presentation and like great summary and everything like that. It's almost like watching like a final presentation. It's really good.
Thank you, too kind.
That's right.
But yeah, well, one of the limitations that we actually have on the chip data set that we're using is there isn't going to be much variation between the actual cases that we'll be considering, because most of them have been treated in very similar ways, and the main difference they're going to have in terms of their anatomy is it's only going to vary very slightly with the registration and slight change of the anatomy. So it's going to be very hard to kind of be able to test the case, like you said, with
right. So could you test if? So I thought that would have a lot of variation. So if you're kind of looking at applying this model in a wider sense, so not just for this data set but other data sets, is there any way to kind of test that variation? So really different patients. Just get another data set. Could you modify them? Like what? How do you go about that?
So one of the ways that we could do it is we could deform some of the patients in there and make deformations, and that could be used to kind of test the sensitivity of the framework in how it compares with very drastic differences in the probability maps being used. So yeah, taking into the account and still using the data set, but just making a couple of morphological changes of it to be able to simulate.
Manually changing it. Okay, and then as like almost like a sanity check, like how, I don't know if you saw Broderick's, but he did some, extreme manual changes just to see if it's affecting it in the way that you'd expect it to, that kind of thing.
Yeah, very similar. Yeah.
Yeah, looks really good, man. It looks like a lot of work.
Thank you very much, though.
Yeah, that's fine. Thanks, Andrew. Any other questions from Kyle? Any questions? All clear? A lot of work, Kyle. All the best.
I'll just add a comment, not a question. It was great, Kyle. Really well done. You obviously understand all the background really well. Just something I didn't actually tell you that would have helped you answer Andrew's question.
Okay.
Just teasing you. When the 5 patients we actually got from a larger data set, and we chose those 5 by clustering a very large trial data set. We had 750 patients, and we clustered them and found the 5 patients that best represented the diversity in that population.
Gotcha. Okay.
Right.
Yeah.
Okay.
Yeah, so that's I forgot to mention that you're armed with that knowledge now.
Yep, I'm writing that down.
Yeah.
Thank you very much.
Any other questions, comments, guys? So good. So great, well done.
Maybe
Ryan.
can I jump in
Yes,
quickly?
Jake.
Yeah, sure.
Jake.
It's just a question about sort of time management, I guess. I wanted to say, first off, again, really, really good presentation. But I think that's a lot of work to get through in a master's project, at least from what you presented today.
Yeah.
Do you anticipate that you'll be able to get through all that in the 1.5 or 2 years that you've got?
In all honesty, not entirely sure.
Yeah,
We'll
fair
absolutely do the best, but yeah, hopefully within the first couple of months be able to get a better idea, and if required, may have to pull back the original scale of the project. So
yeah,
but yeah, and even consideration of putting that through to maybe a PhD or something like that. But yeah, definitely a lot. Yeah.
Yeah, I was going to say it almost seems like the scale of a PhD project, with the aims that you've presented there. So yeah, really good work. Thanks, Carl.
Yeah, thank you.
That's right. Yes, that's great. Brilliant. Thanks very much, everyone. So, Roderick, if you don't mind, if you can stop recording.